everyone, this will be my first video in English, but let me introduce myself first. I am a third year genetics, molecular biology and genetics student at Acıbadem University. Next year will be my last year, so I feel like I can talk about things that interest me and fix some misconceptions about especially biology in social media. And for my first video, I thought that I should do a topic that I'm already familiar with, so then that I can explain it better, and it will help me study as well. So let's start with our first video, Immunology. Welcome to the world of Immunology. Let's start. Have you ever thought about why you weren't sick 24/7? We live in a world teeming with millions of invisible invaders, but we are still standing. Welcome to the first episode of our immunology series. Today we are going back to the 18th century to see how we discovered the invisible fortress inside us. In 1796, an English physician named Edward Jenner played a game of biological connect the dots. He noticed that milkmaids who caught a mild disease from cows called cowpox never caught the deadly smallpox. He realized that the exposure to weak enemy could train the body to fight the strong one. This was the birth of vaccination. Fun fact, the word vaccine comes from the Latin word vacca, which means cow. By the late 1800s, the scientific community was split into two teams. One team, led by scientists like von Behring, believed our protection came from the humors, the liquid part of our blood where antibodies live. The other team, led by Ali Mechnikov, saw actual cells eating germs under the microscope. He called them macrophages. Who was right? Well, it turns out our body uses both a liquid weapon and a cellular army. But where do these soldiers come from? It all starts in your bone marrow with a single legendary cell, the homeopathic stem cell, HSC. I like to call it the mother cell. It has the power to become any blood cell you need. It splits into two lineages. First, myeloid line, which builds your fast-acting innate responders. Secondly, lymphoid line, which builds your high-tech adaptive special forces, the T and the B cells. The innate system doesn't see flow or strip. It sees patterns. PAMPs, pathogen-associated molecular patterns, essential structures of microbes that cannot be mutated easily. Example, LPS in gram-negative bacteria, peptidoglycan, or double-stranded RNA. PRRs, pattern recognition receptors, the host sensors. TLRs, tool-like receptors. Now we are following the myeloid lineage. These are your body's first responders. They don't need a formal introduction to the enemy. They are born ready to fight. But for my fellow students watching, here's a golden exam point. While most of these cells come from the bone marrow, some of our tissue-resident guards, like the microglia in our brain, actually come from the yolk sac before we were even born. They are the three original residents. First up, let's meet the macrophages. They start as monocytes in your blood. But when they enter the tissue, they graduate into these long-lived massive eaters. They don't just eat pathogens, they clear out the trash in your own dead cells. Then we have the neutrophils. Think of them as the kamikaze plots of the immune system. They are the most abundant white blood cells, but they only live for a few days. They rush to the infection site with massive numbers, release their toxic granules, and die in the process. If you've ever seen a pus in a wound, you are basically looking at the battlefield of a fallen neutrophil. But who tells the rest of the army what's happening? This is the dendritic cell. These cells use a process called micropinocytosis. They literally drink the extracellular fluid of the sample to what's inside. They are the spies that bridge the gap between innate and adaptive immune response. They take a piece of the enemy and travel to the lymph nodes to present it to the T cells. Now, how does a cell without eyes know a bacteria is a bacteria? It's all about patterns. Bacteria and viruses have signatures called PAMPs, pathogenic associated molecular patterns, that haven't changed in millions of years. Our cells have sensors called PRRs to detect them. If you're studying for exam, memorize that TLR4, it lives on the cell surface and detects LPS from gram-negative bacteria. When it's triggered, it activates a master switch called NF-kappa-B, which tells the cell to start pumping out inflammatory cytokines. If the threat is still inside the cytoplasm, the NLRs or node-like receptors take over, forming a complex called the inflammasome. But there's a fascinating question. Does every foreign thing trigger this response? Look at this clinical case of argyria. This man's skin turned blue because of silver deposits. He is technically full of foreign material, yet he has no inflammation. Why? Because silver is inert. It doesn't have PAMPs. It doesn't trigger the PRRs. This teaches us that the immune system isn't just looking for the foreign objects, it's looking for dangerous patterns. No PAMPs, no PRR activity. No PAMPs, no PRR activation, no fire alarm. When the fire alarm is pulled, we see inflammation. For your example and for mine, remember the 4 Latin pillars of inflammation: rubor, redness, color, heat, tumor, swelling, and dolor, pain. Mechanically, it's a beautiful process. Cytokinesis like TNF-alpha make the blood vessels leaky. This allows fluid and troops to squeeze through the vessel walls following a chemokine gradient. Specifically, CXCL8, which acts like a GPS for the troops. So, from a blue skin to leaking blood vessels, the innate system is like a masterpiece of biological engineering. It's the difference between a minor scratch and a fatal infection. I'm going back to my signaling pathways now. I still have to master the complement system. Stay sharp. I'll see you in part 2, where we meet the special forces, T and B cells. Welcome back to part 2. If the innate system is the frontline infantry, adaptive immunity is the strategic special forces. While the innate response starts in minutes, the adaptive response takes days to mobilize. But when it activates, it is laser focused and incredibly powerful. This part, we meet the B and the T lymphocytes. The secret to adaptive immunity lies in antigen receptors. B cells bear the B cell receptor BCR, which is essentially a membrane-bound antibody. They can see antigens in their native form, floating in the blood or lymph. T cells, however, are more sophisticated. Their T cell receptor, TCR, is blind to free-floating antigens. They only recognize antigen fragments presented on MHC molecules. Exam quick note. Remember the math for your exams. CD8 T cells pair with MHC class 1. 1 times 8 equals to 8. To kill infected cells, CD4 T cells pair with MHC class 2. 4 times 2 equals to 8. To act as helper cells, coordinating the entire response. If the math doesn't equal to 8, the molecular handshake won't happen. So, let's solve a biological mystery. We only have about 25,000 genes, yet we can recognize over 100 million different pathogens. How? During development, lymphocytes pick gene segments called variable diversity and joining segments and snap them together like a Lego bricks. This is combinatorial diversity. But then, it gets even better. When these segments join, random nucleotides are added and removed at the joints. This is junctional diversity. This messy process ensures that you have a receptor for almost every possible shape in the universe. Once these receptors are made, we use McFarlane-Burnett's clonal selection theory. We start with millions of unique naive lymphocytes. When a pathogen enters, it selects one of the matching receptors. The cell doesn't just sit there, it undergoes clonal expansion, dividing rapidly to create an army of thousands of identical clones. Note for exam, remember this process is irreversible. Once a cell rearranges its DNA, all its children inherit the specific identity forever. With all this shuffling, what stops us from attacking our own cells? It's tolerance. During their education in the bone marrow and the thymus, any immature lymphocyte that reacts too strongly to our own self-antigens is forced to undergo apoptosis, programmed cell death. This is clonal deletion. If this fails, we get autoimmune diseases, where the fortress attacks itself. Finally, the superpower of the adaptive immune system. Memory. Look at this graph. The primary response is slow, but there's a lag phase while the body searches for that 1 in a million cell. But after the infection is cleared, we keep our memory cells. In the secondary response, there is no lag. The response is faster, stronger, and in the case of B cells, the antibodies bind even tighter. This is called affinity maturation. It is a biological proof that what doesn't kill us actually makes us immune. From the basic Lego logic of our DNA to recall power of our memory cells, the adaptive system is the peak of our biological engineering. I'm heading back to do my studies. Thank you for being with me in this part, and I'll keep the videos coming for you. Everything that I mentioned in this video will be put in the description box below. Please don't forget to subscribe and hit the thumbs up button. See you in the next one.